species. Many thanks to the organizers for the invitation to participate in this symposium. It is really my pleasure to tell you a little bit about the research that we do in our Plant Evo research group. This group was established like 5 years ago as a collaboration between the National University of Colombia here in Bogotá and the Universidad de Antioquia in Medellín. Our main research topics are focused on the study of the genetic basis of several developmental processes in tropical non-model plants, mainly related to flower and fruit evolution. Although species number is lower in early divergent lineages, we have focused on them because their morphological variations are in fact higher and somehow less understood, given that the majority of model plants are less variable, more predictable, and better understood. As I will tell you in this presentation, we have integrated several techniques, including virus-induced gene silencing, in situ hybridization, protein-protein interactions, and a large doses of transcriptomics. Let's start the talk by saying that the flowering plants have undergone at least 7 whole genome duplication events, here marked with the yellow stars, that have been critical for their evolution and diversification. For instance, our breakfast today is nothing else than a bouquet of polyploids. However, as I mentioned before, we are primarily interested in early divergent lineages that are not represented by the model genomes or systems for functional analysis, and those that predate the major whole genome duplication events in flowering plants. Our research group is interested in addressing general questions in evolutionary developmental biology. For instance, one of these questions ¿Más bajo? Más despacio. Primera vez que me pasa en inglés, perfecto, mucho mejor. ¿Me devuelvo o no? Uy. Okay, let's start here again by saying that flowering plants have undergone at least 7 whole genome duplication events. Here, you can see them with the yellow stars in the cladogram, which have been critical for their evolution and diversification. For instance, our breakfast, I told you, is a large collection of polyploid plants. However, our main interest is in groups that predate those whole duplication events in the genome. For instance, one of the questions is why plants such as the early divergent genus Aristolochia, which you see here, have taken on such amazing floral morphology variation, perhaps more so than any other group of flowering plants. Aristolochia comprises about 600 species with an amazing floral variation, not only in terms of floral shape, size, color, but also in terms of epidermal elaborations that are unique in the plant kingdom. Thus, we have focused. Can we play the video here? On the so-called matchbox genes, those that are genes responsible for floral organ identity as a proxy to understand the evolution of these flowers. Aristolochia perianth is differentiated into 3 portions, an inflated one at the base called the utricle, followed by a narrow portion named the tube, and an apical portion which is called the limb. This last one expands at maturity basically to seduce pollinators. Colombia has the smallest and the largest Aristolochia flowers worldwide, varying from about 1 centimeter in the tiny Aristolochia numularifolia in the Orinoco Plains, by the way, collected for the first time by Humboldt and Bonpland, to flowers of about 2 meters long in Aristolochia grandiflora, which inhabits Chocó and neighboring areas. Despite this extreme size variation, the cell division, expansion, and differential growth are genetically regulated by the same genes, some of which have actually not been reported in driving floral symmetry or perianth identity elsewhere. We have applied in situ hybridization to closely examine such genes. This is a technique that allows to detect where and when a gene is turned on, the purple areas in the slides. Here I am showing you the expression of matchbox genes responsible for floral organ identity in Aristolochia fimbriata. Whereas the B and C class genes important for stamen and carpel identity in Aristolochia fit the model predicted from Arabidopsis, we found little conservation on the genes that are important for perianth identity, as they have broad expression patterns very different to what the model predicts. The good news is that we found an alternative, non-canonical gene that appears to be responsible for the identity of these bizarre flowers. This is in fact the gene agamus-like 6, also known as AGL6, which so far has never been associated with perianth identity. In addition, by implementing a protein-protein interaction method using yeast to hybrid, we were able to identify that the formation of an AGL6 sepalata tetramer is in fact required to establish perianth identity in Aristolochia. The second question we are addressing at the moment is the control of the bilateral symmetry in flowers. This trait is critical to successful pollination. Now we know that bilateral symmetry in Aristolochia flowers follow a different gene mechanism to that known to occur in, for example, orchids or snapdragons, which have been the model to study bilateral symmetry. In green, I am showing you the TCP transcription factors that are important for reducing cell proliferation on one side of the flower with respect to the other. We, in fact, have been able to find that genes known as cincinata genes, which are also from the TCP family but are not the canonical cycloidea genes from the model species, are responsible for such bilateral symmetry in Aristolochia flowers. And again, this is a function that has not been reported before. Our question number 3 has to do with the genetic mechanisms shaping the different portions of the flower. We have also studied differential gene expression in the 3 floral portions, whereas the limb and the tube have specialized hairs to trap the insects. The utricle is covered by nectar-secreting hairs to feed the trapped pollinators. For this, we generated individual transcriptomes from each portion of the perianth. Through this approach, we were able to identify the top 20 genes differentially expressed between the limb, the tube, and the utricle. Among them, cincinata genes also appear to play a role in cell proliferation. We have actually been able to find also a microRNA 319, which is a post-transcriptional regulator of the gene cincinata 1, and we have validated this finding via real-time PCR. Now, let's move to the fruit projects we have. Fruit development and evolution is as diverse and complex as flowers. We aim to study the genetic mechanisms that drive fruit development and evolution in 3 main groups of flowering plants: the poppy family, the tomato family, and the coffee family, three of the most important groups in terms of economic botany. In these three unrelated families, fleshy fruits evolved from dry fruits. And what we are asking is whether there are similar patterns or similar genetic mechanisms regulating these shifts. We hope to have some answers for our next binational meeting, hopefully. So far, we can anticipate that there are some genes required in all three case studies, but are others that appear to be species-specific. I'm going to show you one example in the tomato family. Here we have applied a technique called virus-induced gene silencing. This is a powerful technique that allows direct functional analysis. This technique takes advantage of the plant defense response to viruses. The target gene is inserted into a viral vector that in turn is inserted into agrobacterium, which is used to infiltrate the plant. The plant actually responds by silencing the inserted gene, thinking that she recognizes the gene as part of the virus, not a gene. So the transformed plant shows a mutant phenotype, which is the result of the down-regulation of the target gene. Given that this transformation is transient, this is not fixed in the DNA at all. This is at the RNA level. The phenotype is not to be considered a genetically modified organism. By using VIGS, we have been able to identify transcription factors that are important to stop lignification in both dry and fleshy fruits. When we silence these genes, we in fact get more lignification in the fruits. Finally, I will finish with the plant parasitism. Our interest in this topic is to examine the evolution of the plastids. That is, bacteria evolutionarily trapped since a long time ago in the plant cytoplasm. Thus, they are the microbiomes of the plant cells, which are, by the way, responsible, among other things, for the production of oxygen on Earth. We are studying plastome evolution at three levels of parasitism, from facultative parasites to my right, to holoparasites to the left. So far, we have sequenced and assembled the plastome of Castilleja paramensis, this tiny, tiny plant from the paramos, and the holoparasitic Pilostyles boyacensis, which grows around Bogota in very dry areas of the Cordillera Oriental of Colombia. The genus Castilleja was already included by Humboldt as a paramo element in his famous Geography of Plant chart. Our Castilleja paramensis can grow free or associated underground to another paramo endemic, Plantago rigida. Sorry. They can communicate by little haustoria between them and grow together, but Castilleja paramensis can also grow freely. Surprisingly, the plastome, which is the chromosome of the chloroplast, is actually larger in this plant than that of the, for example, Arabidopsis or other non-parasitic plants. That is an open question. Why is larger than non-parasitic plants? We still don't know the answer. On the contrary, the plastome of the holoparasite Pilostyles boyacensis, as expected, is very much reduced. Not only 7 genes, to only 7 genes, and over 12,000 base pairs, being one of the most reduced plastomes across the plant kingdom. This makes sense, as our paramo ecosystems are quite young, whereas similar extreme plastome reduction in pilostyles has been reported from African and Australian species, from which we can assume that this plant parasitism is actually one of the oldest on Earth. With this, I finish my presentation. These are the founding sources we have had in the 5 years of existence. And this is the happy family that have helped a lot in the projects we have at the moment. People from all over the world, New York, Valencia in Spain, Germany, and the rest are more or less Colombians. I just want to finish with this. This is Dr. Natalia Pavón Mora, who is the associate professor at the University of Antioquia. She is the co-leader of the group, and she says hello from Dresden, where she is now a visiting professor at the moment. Thank you very much.